Hello, I'm Sarah, one of the presenters of this video. In this video, we will talk about fructose metabolism that happen in organs in ourselves. Fructose metabolism occurs in the liver, intestine, and kidney of human for major pathway. For minor pathway, fructose metabolism occurs in adipose tissue and muscle.
Alright, now we are going to continue to fructose function. Firstly, fructose enhances the activation of pyruvate kinase and thus leads to an increased flux of pyruvate into the Krebs cycle. Then, fructose promotes the uptake and storage of glucose in the liver.
Fructose gives benefit to individual who's doing exercise, especially in athletes, because it provides additional energy to contract skeletal muscle in the form of lactate. Fructose is metabolized to pyruvate more rapidly than glucose, because not through the energy investment step.
Unlike glucose, which actually has its own catabolic pathway in our cells, this sugar, which is fructose, do not have their own individual breakdown pathways. So when these monosaccharides actually make their way into the cells of our body, this sugar must be transformed into molecules, into metabolites that are part of that glycolytic pathway. Once that's happened, we can basically incorporate those glycolytic metabolites into the glycolytic pathway. Now, depending in what type of cell that fructose actually ends up, it can be converted into glycolytic metabolites in one of two ways. Now, let's begin by discussing the fructose 1-phosphate pathway. Fructose 1-phosphate actually consists of three individual steps. Ultimately, what we want to do in this pathway is transform a fructose molecule into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, because these are two molecules that aren't fat glycolytic metabolites, and so we can feed them directly into the glycolytic pathway, and then we can use those molecules to form ATP molecule. So let's see what these three steps actually are. In step 1 of the fructose 1-phosphate pathway, we have an enzyme we call fructokinase. This enzyme basically uses the ATP to phosphorylate carbon number 1 in fructose, and it will form fructose 1-phosphate, also the ATP. Now, what is the purpose of this step number 1? The purpose is to basically destabilize fructose 1-phosphate molecule. Once we add the phosphoryl group that destabilizes our molecule, now the next step is we can basically cleave that fructose by the activity of enzyme we call aldolase B. Aldolase B is an enzyme that catalyzes an aldol cleavage that will form two 3-carbon molecules. One of these is dihydroxyacetone phosphate, DHAP, and this molecule is part of the glycolytic pathway. So, now we take this molecule and replace it into stage 2 of glycolysis. In stage 2 of glycolysis, dihydroxyacetone phosphate is then transformed into glyceraldehyde 3-phosphate. What about the other 3-carbon molecule, or in other words, the glyceraldehyde? Well, glyceraldehyde is not actually part of that glycolytic pathway, but it's very close to glyceraldehyde 3-phosphate. The only thing it's missing is a phosphoryl group on carbon number 3. So all we have to do in step number 3 is use a special enzyme and the ATP molecule to basically attach the phosphoryl group onto carbon number 3, and that's exactly what thiokinase actually does. Now, thio simply means the substrate molecule is a 3-carbon sugar. So we have an attachment of the phosphoryl that we take from ATP and place it onto carbon number 3 that will form our glyceraldehyde 3-phosphate, which can be fed into stage 3 of glycolysis that will eventually converted into pyruvate molecules and ATP molecules. So this is what our fructose 1-phosphate pathway is like, and this is the pathway that is followed by liver cells. So if fructose make its way into the cells of our liver, this is how it's going to be basically transformed into molecules that can be fed into that glycolytic pathway.
Now, in other tissue and other cell, there are simple pathway that is followed. So if we recall stage 1 of glycolysis, we actually form fructose 6-phosphate in step number 2. And in step number 3 of stage 1, we basically transform that fructose 6-phosphate into fructose 1,6-bisphosphate. So the simple step is to actually take that fructose, use an ATP molecule in special enzyme we call hexokinase. and we form fructose 6-phosphate that can be fed directly into the glycolytic pathway in the stage 1. And of course, we form the ADP because we have to use an ATP to actually transfer that phosphoryl group into carbon number 6. So in non-liver cell, other tissue cell of our body, fructose can be converted into fructose 6-phosphate by the action of hexokinase. So these are the two pathways by which the cell of our body can basically incorporate fructose into the glycolytic pathway.
Fructose deficiency occurs due to the deficiency of adelase B, also known as fructose 1-phosphate adelase deficiency. This reduces the cleavage of fructose 1-phosphate by adelase, which also inhibit another enzyme known as fructokinase. The fructose 1-phosphate increase result in the inhibition of both glycolysis and gluconeogenesis, which are responsible for creating glucose. This will lead to the glucose level to being lower than normal level, also known as hypoglycemia.
The accumulation of free fructose in the liver, kidneys, and bloodstream causing a fructose toxicity. The high fructose levels cause an increase in uric acid in the blood known as hyperuricemia, which then leads to gout. Kidneys attempt to excrete the excess uric acid, and this compromises lactic acid excretion. This causes a build-up of lactic acid in the blood above the normal levels, known as lactic acidemia.